Phage typing. Bacteriophages are specific in action. A particular phage may be very specific that will infect only a few strains of bacterial species. On the extreme, another phage may infect strain of two or more species of a particular genus. Susceptibility to lysis by a particular phage may be the only apparent phenotypic difference between two bacterial strains and may be only means by which a strain causing an outbreak disease can be recognized. This observation is a basis for phage typing, a procedure for characterizing and detecting bacterial strain by their reaction whether they are susceptible or resistant to various known strains of phage. The phage is sometimes employed in epidemiology to identify and trace the progress of infectious agent. This has been the standard procedure of staphylococcus aureus involved in outbreaks of food poisoning or other infections. A species-specific phage is used routinely in the identification of Bacillus anthracis, causative agent of anthrax. Thus, reproduction of in laboratory animals is not necessary to confirm identification of this organism. In phage typing, the prepared culture of bacteria will be plated to create bacterial lawn. As soon as the surface of agar dried, add 1 drop of different phage on each region. When the phage spots dried, the agar plate will be incubated at 37 degrees Celsius for 18 hours. At the end of the incubation, the agar plate will be read using a magnifying glass through the bottom of the plate. What will be happened when a drop of phage put on a bacterial lawn? First, a phage infects a single cell in a lawn of sensitive growing bacteria. Then, phage reproduces in the cell, typically yielding about 50 progeny phage per infected cell. Next, lysis of the cell releases phage diffuses through the medium and infects adjacent cells. Phage reproduces in this cell, releasing 50 additional progeny phage per infected cells. Lastly, these cells lyse, releasing more phage which can then diffuse outward and infect the surrounding cells. Lysis of cells results in a circular cleaning in the lawn of bacteria. This region of visible lysis is called a plaque. The morphology of the plaque depends upon the phage, the host, and the growth conditions. Usually phage infection is studied in a layer of soft agar, or top agar, which allows the phage to diffuse rapidly. The size of the plaque is proportional to the efficiency of absorption, the length of the latent period, and the burst size of the phage. A diversity of plaque sizes can result if the phages infect cells at different times during the bacterial growth phase. Phage that absorb early make larger plaques than those that absorbed later. To avoid this, phage are often pre-absorbed to cells under conditions that do not allow phage growth. Example, low temperature then shifted to permissive conditions to induce all of the phage to develop at the same time. A clear plaque will be formed if the host is completely susceptible to the phage. Often a clear plaque will be slightly turbid at the edge because the cells at the edge of the plaque are not yet fully lysed. If the host is partially resistant to the phage, then the plaque may be uniformly turbid, for example if 10% of the cells survive phage infection. Why do plaque assays accurately measure the titer of temperate phage? If a phage lysogenizes a host cell immediately upon infection, it would never form a plaque. Instead, when a temperate phage infects a population of exponentially growing cells, each phage produces a plaque with bullseye plaque morphology, a turbid center surrounded by a ring of clearing. This characteristic plaque morphology is due to the role of the MOI or multiplicity of infection and cell physiology on the lysis-lysogeny decision. Lytic growth is favored when cells are growing rapidly and the MOI is low. Lysogeny is favored when cells are growing slowly and the MOI is high. This is why temperate phage typically have blocks with turbid centers. When the cells are initially infected with phage, the ratio of phage to cells is usually about 1 phage per 106 cells. Initially, the nutrients are plentiful so the bacteria grow rapidly, and since the MOI is low, the phage grow lytically. After several lytic cycles, the local MOI increases and most of the cells are lysed, producing a plaque in the lawn of cells. As the cell lawn becomes saturated, the rate of cell growth slows down, and since lysis required rapid metabolism, the plaque stops increasing in size. However, any lysogens that form in the center of the plaque are immune to lysis and can continue to grow since they do not have to compete with nearby cells for nutrients. Thus, lysogens begin to grow in the center of the plaque, giving the plaque a turbid, bull's-eye appearance. Phage susceptibilities will be evaluated by means of the plaque number, size, and transparency. The pattern will be compared with known phage type patterns in the databases and defined. If the culture did not react with any of the typing phages, it was defined as non-typable. And if the culture reacted with typing phages but gave a different pattern other than those in the databases, it will be considered as reacting with the typing phages. But lytic pattern did not correspond to any recognized phage types. So-called RDNC or reacting with the typing phage, but lytic pattern did not correspond to any recognized phage types. What are the advantages of using this procedure? The advantages of using phage typing is that it is reproducibility. Reproducibility means that the technique has ability to yield the same result when the same strain is tested repeatedly. It has a discriminatory power. It is the ability to differentiate among unrelated strains. Ideally, each unrelated isolate is detected as unique. Ease of interpretation. The typing test produces reproducible and unambiguous results that can be interpreted easily. What about its disadvantages? First, it is difficult to perform. Second, it needs well-equipped laboratory for the preservation of virus. This technique requires maintenance of biologically active phages and hence is available only at reference centers. Even for the experienced worker, the technique is demanding. And last, it is not used for all type of microorganism because bacteriophage is highly specific to its host. Many strains are non-typable.